Let's we discuss after pollination in flowering plants. One of the most important event is pollen pistil interaction. Generally, this is one stamen. Each stamen having two major parts, filament or stalk, and fertile part is called as anther. Most of the anthers are bilobed and diathecous, and inside many pollen grains are produced. At mature condition of anther, when anther wall is ruptured, the pollen grains are released. The transfer of this pollen grain from anther to surface of stigma is called as pollination. After pollination, when a pollen grain is land on surface of stigma, when the pollen grain land on surface of stigma, up to here pollination. After pollination, there is an interaction between pollen grain and stigma is happened, that is called as pollen pistil interaction. So generally, pollination is not ensured to reach the pollen grains on the surface of stigma. Generally, the stigma receives two different types of pollen grains. One is right pollen grain and another one is wrong pollen grains. After pollination, if the stigma is receives two different types or both types of pollen grains, one is right pollen grain, another one is wrong pollen grain. The right pollen grain is called as compatible pollen grains and wrong pollen grain is called as incompatible pollen grains. Means after pollination, the stigma receives two different types of pollen grains, right pollen grain and wrong pollen grain. Right pollen grain is called compatible pollen grain, wrong pollen grain is called incompatible pollen grain. Generally, according to self-incompatibility, there is a chemical dialogue is happened in between pollen grains and stigma of pistil. The wrong pollen grain is rejected and the right pollen grain is accepted. This is called as chemical dialogue or chemical dialogue. When a chemical dialogue is happened, that means the process of rejection and acceptance. When the stigma is accepted pollen grain, if the landing pollen grain is right pollen grain or compatible pollen grain, there is a chemical dialogue acceptance. If the stigma receives wrong pollen grain or incompatible pollen grain, there is a chemical dialogue rejection. The process of acceptance and rejections is nothing but chemical dialogue in between pollen grains and stigma. When the compatible pollen grains germinate and produce pollen tube, for example, wrong pollen grains are rejected, right pollen grains are accepted. When the right pollen grain or compatible pollen grain is accepted, then the pollen pistil interaction is started. Then the stigma provide water and sugars. Then by using water and sugars, the pollen grain size will be increases and the exine will ruptures, the intine produces pollen tube. Here I am explaining germination of pollen grain on surface of stigma. When the exine is ruptured, the intine produces pollen tube. Then the largest cell in a pollen grain is in two-celled pollen grain, vegetative cell or tube cell with dense cytoplasm. The vegetative cell or tube nucleus, it is occupies at the tip of pollen tube. Then next, the generative cell enters into pollen tube, undergoes mitotic division to produces two amoeboid shaped haploid male gametes. This is male gametophyte development. And it is having callose plex. This is the process of germination of pollen grain on the surface of stigma and formation of pollen tube from the intine, and all the components of pollen grains enters into pollen tube. When the pollen tube is formed, this pollen tube digests the tissue of stigma and digests the tissue of style and this pollen tube reaches to style and reaches to ovary with two male gametes. When the compatible pollen grain accepted on the surface of stigma, there is a germination of pollen grain on the surface of stigma takes place. Pollen grain germination can take place. Digestion of stigma tissues and style tissues, the pollen tube enters into style and enters into ovary. And the next, after reaching the pollen tube into ovary, then the pollen tube trying to enter into ovule. The entry of pollen tube into ovule by three different ways. The first way of entry of pollen tube into ovule is porogamy. The second way of entry of pollen tube into ovule is chalazogamy. The third way of entry of pollen tube into ovule is mesogamy. Once again, I repeat from introduction, pollen-pistil interaction. After pollination, when the stigma receives right pollen grain and wrong pollen grain, right pollen grain is called compatible pollen grain, wrong pollen grain is called incompatible pollen grain. When the stigma receives right and wrong pollen grain, compatible and incompatible pollen grain, incompatible pollen grains are rejected and compatible pollen grains are accepted. This is a chemical dialogue in between right pollen grains and stigma of pistil. When the compatible pollen grains started to germinate, it produces pollen tube. This is pollen grain germination. The germinated pollen grain produces pollen tube. The pollen tube enters into the style through the digestion of tissue of stigma and the tissues of style, and pollen tube reaches to ovary and pollen tube try to enter into ovule. The entry of pollen tube into ovule with two male gametes. There are three ways entry of pollen tube into ovule. One is porogamy, chalazogamy, mesogamy. So, out of these three different types of entry of pollen tube into ovule is of three types. Out of these three types, the most common type of entry of pollen tube into ovule in flowering plants or angiosperms is porogamy. Porogamy means the entry of pollen tube into ovule through the micropyle. Once again, this is called as micropyle. The terminal part of the terminal opening pore in the structure of ovule is called micropyle, where the integuments are leaving free. The entry of pollen tube into ovule through the micropyle is called as porogamy. It is the most common type amongst flowering plants. The second type of entry of pollen tube into ovule is chalazogamy. The process of chalazogamy, it was first discovered by one of the scientists. His name is Trube. The scientist Trube, in the plant of Casuarina, he was first discovered chalazogamy. Chalazogamy means the entry of pollen tube into ovule through the chalaza, the base of the ovule. Opposite to micropyle, the base of the ovule is called as chalaza. The entry of ovule into the entry of pollen tube into ovule through the chalaza is called chalazogamy. It was first discovered by Trube in the plant of Casuarina. The last and final most method of entry of pollen tube into ovule is mesogamy. Means the entry of pollen tube into ovule through the integuments, the protective coats. The protective coats of ovule is called integuments. The entry of pollen tube into ovule through the integuments is called as mesogamy. The mesogamy commonly seen in cucurbita plants. Out of these three entry of pollen tubes into ovule, the most common is porogamy. It is the most common amongst these three types in angiosperms or flowering plants. When the pollen tube enters into ovule through the micropyle, by the process of porogamy. The next pollen tube enters into embryo sac. Inside the ovule, embryo sac is present. When the pollen grain is germinated, pollen tube enters into style, pollen tube enters into ovary, pollen tube enters into ovule, pollen tube next enters into embryo sac. Let's discuss entry of pollen tube into embryo sac or female gametophyte. Entry of pollen tube into embryo sac. Generally, in pollen pistil interaction, the simplest definition, the germination of pollen grain on the surface of stigma. Germination of pollen grain. Germination of pollen grain on the surface of stigma. 2. Entry of pollen tube into ovule. In between these, whatever the steps are takes place, that phenomenon is called as pollen pistil interaction. The steps can take place in between the germination of pollen grain on surface of stigma and entry of pollen tube into ovule. In between these is called as pollen pistil interaction. When the pollen tube with 2 male gametes enters into ovule, out of 3 pathways for entry of pollen tube into ovule, the most common is porogamy, means entry of pollen tube into ovule through micropyle. After reaching pollen tube into ovule, the next pollen tube enters into embryo sac. In angiosperms, embryo sac is also called as female gametophyte. The embryo sac or female gametophyte in angiosperms is 7-celled and 8 nucleated. So, whenever the pollen tube enters into embryo sac, ovule, the next it is trying to enters into embryo sac. Before that, let's discuss one of the very most important point in this concept. When a right pollen grain or compatible pollen grain land on the surface of stigma, there is a great signals are transferred towards embryo sac in egg apparatus. 2 synergids are present and 1 egg cell is present. When right pollen grain land on the surface of stigma, when compatible pollen grain start to germinate on surface of stigma, out of 2 synergid, 1 synergid will be degenerated. And it has filiform apparatus. The recent studies, what it reveals, the filiform apparatus, it can be guide the entry of pollen tube into embryo sac. Now here we discuss how the pollen tube enters into embryo sac. Generally, this is the pollen tube. The pollen tube, it carries 2 haploid amoeboid shaped male gametes. After entry of pollen tube into ovule, the next step entry of pollen tube into embryo sac. The entry of pollen tube into embryo sac through the degenerated synergid. This pollen tube is attracted by filiform apparatus. The attracting pollen tube by filiform apparatus is called as chemotactic. Now, after entry of pollen tube into embryo sac through the degenerated synergid, the next step here, entry of pollen tube into embryo sac through the degenerated synergid. It has 2 male gametes and the tip having tube nucleus or vegetative nucleus with dense cytoplasm. The tube nucleus will be degenerated. At the tip of these pollen tube, there is a small pore-like structure is formed. Through this pore, these male gametes releases into embryo sac. 2 male gametes releases into embryo sac through the pore at the tip of pollen tube. The first male gamete, haploid male gamete, reaches to haploid female gamete is also called as egg cell. When the egg cell is also called as female gamete, ploid is haploid, ready to crosses with haploid male gamete. The process of fusion of haploid male gamete with haploid female gamete, this process is called as fertilization. Fertilization is also called as syngamy. And the haploid female gamete and haploid male gamete produces a diploid zygote. One of the scientists, his name is Strasburger. He was first to discover fertilization or syngamy in the plant of Monotropa. In the plant of Monotropa, the scientist Strasburger, he was first to discover fertilization or syngamy, then its form zygote. The second haploid male gamete, it's moved towards central cell in embryo sac. The central cell embryo sac having two polar nuclei or diploid secondary nucleus. Now this diploid secondary nucleus combines with one haploid male gamete by the process of triple fusion. Why it is called as triple fusion? The secondary cell or central cell having two polar nuclei, one male gamete nuclei. Total 3 nuclei participated in this process, then it is called as triple fusion, then it produces triploid primary endosperm nucleus, or it is also called as PEN. That process of triple fusion was first discovered by one of the scientists, his name is Navachin. The scientist Navachin, he was first to discover triple fusion in the plant of Fritillaria. In the plant of Fritillaria, the process of triple fusion was first discovered by Navachin. And here, inside the embryo sac or female gametophyte, 2 different types of fertilization can take place. Here syngamy and here triple fusion. Both syngamy and plus triple fusion is called as double fertilization. Exhibit double fertilization is the characteristic features of angiosperms. Double fertilization is the characteristic features of angiosperms. Double fertilization means syngamy plus triple fusion is equal to double fertilization. Double fertilization also was first discovered by Navachin. Here the true fertilization is syngamy, the vegetative fertilization is triple fusion. At the end of double fertilization, the product is zygote and primary endosperm nucleus. Zygote and primary endosperm nucleus, these are the products of double fertilization. Double fertilization is the characteristic features of flowering plants or angiosperms. Pollen germination we can made inside the lab. Generally, whatever I discussed today, this topic is a major experiment for second year biology lab. Generally, pollination takes place in a flowering plants naturally. Pollen germination takes place in a flowering plants naturally. But pollen germination we can create artificially inside the lab. The germination of pollen grains in vitro, that means inside the lab, is called as in vitro pollen germination, is also called as hanging drop experiment. Why this experiment is called as in vitro pollen germination or hanging drop experiment we discuss now. For this experiment inside the lab we required very clean and clear glass slide. This is the first requirement. And the second requirement is 10% of sugar solution. 10% of sugar solution means 10 grams of sugar present in 100 ml of solution. 10 grams of sugar in 100 milliliters of solution is called as 10% of sugar solution. So, we are preparing inside the lab 10% of sugar solution and we are preparing very clean and clear glass slide. and we are collected a chemical substance as boric acid. It is having antiseptic properties, and we are collected nutrients like calcium, magnesium and potassium. And for this experiment we need a flower. A common flowers we are used for in vitro pollen germination or hanging drop experiment or artificial pollen germination, such as Vinca rosea, Crotalaria, pea plant flowers, chickpea flowers and balsam flowers. In biolab, for in vitro pollen germination or hanging drop experiment, the most commonly used flowers are Vinca rosea flower. This is a Vinca rosea bisexual flower. This bisexual flower having male sex organ is called as stamens. Each stamen having two parts, filament or stalk, and another one is anther. Inside the anther there is a pollen grains are developed. From this Vinca rosea bisexual flower, the stamens are separated, and with the help of sharp needle from these anther we are making rupture, then we are dusted pollen grains. Before the dusting of these pollen grains, with the help of a dropper, we are collecting a drops of 10% of sugar solution with the help of dropper, and we are keeping a drop of sugar solution, 10% sugar solution on the clean and clear slide, and later rupture of anther and dusting of pollen grains in this drop of 10% of sugar solution. And later we keep a thin cover slips on this drop 10% sugar solution and dusted pollen grains. We are keeping this entire experimental setup 15 to 30 minutes duration under compound microscope or under simple microscope. If you can clearly observe in this 10% of sugar solution, each and every pollen grain, it shows an outgrowth. This outgrowth is nothing but pollen germination. So the pollen germination we are created inside the lab artificially with the help of this experimental setup, then it is called as in vitro pollen germination, and 10% of sugar solution we are keep on the glass slide, then it is called as hanging drop experiment. This is a method of artificial pollen germination or hanging drop experiment. In the concept of fertilization, one more method to production of a desirable offsprings with superior characters. Superior characters in the sense resistant to insects and pests, resistant to unfavorable conditions, and production of more crop productivity. All these are desirable characters. A production of new desirable plant species, it's happened even inside the lab by the process of artificial hybridization. In this class, we discuss artificial hybridization. In artificial hybridization, before going to that, what is hybridization we discuss. The definition for hybridization is the crossing between two genetically different parents is called as hybridization. For example, this is one genetically different parent and this is one genetically different parent. The crossing in between two genetically different parents is called as hybridization. If this hybridization is happened inside the lab is called as artificial hybridization. And after artificial hybridization, a new offspring is produced. That new offspring having both the parents' desirable characters. The incorporation of desirable characters into next generation or offsprings by the process of artificial hybridization. The hybridization is happen maybe in between two different varieties, or maybe two different species, or maybe two different genera. Hybridization is happen in between two different species or two different genera or two different varieties. Reason is to incorporate desirable characters into next offsprings or next generation. That means artificial hybridization is one of the best crop improvement program. The best crop improvement program is artificial hybridization to incorporation or insertion of desirable characters from two different varieties or two different species or two different genera into next offsprings. The entire process of this artificial hybridization, it depends on 4 different steps, such as the first one is selection of parents. Second one is emasculation. Third one is bagging. Fourth one is artificial pollination. For example, this is one genetically different species. This is one genetically different species. This species having bisexual flowers, and this species stamens and anthers produces pollen grains. These pollen grains are having desirable characters. Desirable characters means resistant to environmental conditions and insects and pests. I want to transfer these desirable pollen grains in between these two genetically different species from this anthers to this stigma artificially inside the lab. For this procedure, for artificial hybridization, it requires 4 different steps. The first one is selection of parents, second one is emasculation, third one is bagging, fourth one is artificial pollination. The first step, very most important step in artificial hybridization. Selection of 2 genetically different species. As a parent, it's very important, toughest task. The healthiest parent selection is important. If you are selected parents, very weak parents, the next generation is very weak. It's not having any desirable characters. It is not resistant to pest and insect and environment. Crop productivity will decreases. So that's why in artificial hybridization, selection of parents is foremost important characters. Selection of desirable, healthiest parents is called as selection of parents. If the selected parent having the flowers is bisexual, there is a chances of transfer of pollen grains from anther to surface of stigma. If the same pollen grains land on same surface of stigma, due to continuous self-pollination, the genetic diversity is limited and inbreeding depression is happened. There is no new variation in the next generation. So that's why before the rupture of these anthers, before maturation of anthers, the removal of stamens from the bisexual flower for prevention of self-pollination is called as emasculation. Emasculation means the process of removal of stamens from bisexual flowers is called as emasculation. Emasculation prevents self-pollination in a bisexual flower in selected parents. Now this is a unisexual female flower. For prevent unwanted pollinations, these unisexual female flowers, it is covered by a polythene bag, must be prepared by butter paper. The unisexual female flower, it is covered by a polythene bag, it is prepared by butter paper. This phenomenon is called as bagging. Bagging prevents unwanted pollination. And last and final most step is artificial pollination. Already inside the lab from other desirable plant with desirable pollen grains, we are selected with the help of 4 ships. With a sterile condition, this polythene bag will be removed inside the lab and the transfer of these desirable pollen grains on the surface of stigma inside the lab by the plant breeders. This is called as artificial pollination. When the desired pollen grains we are keep on surface of stigma, again the polythene bag will be covered. This process is called as rebagging. And when the desirable pollen grains land on surface of stigma, then pollen pistil interaction is happened, pollen grain germination takes place, and pollen tube growth can take place towards style and reaches to ovary and reaches to ovule, and fertilization takes place to form zygote and endosperm, and finally it forms seed. This seed produces new generation. That generation plant is resistant to environmental conditions and resistant to insect and pest, and production of more crop productivity. So, the incorporation of desirable characters into next offsprings by the method of artificial hybridization. If the selected parent is unisexual, that means both the sex organs are not present, then emasculation is not required. Emasculation is required for bisexual flowers, not required for unisexual flower. It is one of the best plant breeding method to increase the crop productivity, to increase the desirable characters in the next offsprings.